Okay, so welcome back to this next video in which we are discussing cholesterol metabolism. Okay, right, so I've put up this picture which is our overview of cholesterol metabolism so that we can look at which portions of this pathway that we've actually discussed now. So we've discussed how there is this store of cholesterol within the blood in the form of LDL particles, which is known as the LDL pool. And we've discussed now how cholesterol poor cells, so either cholesterol poor peripheral cells or cholesterol poor hepatocytes, can remove cholesterol from this LDL pool through that LDL receptor mediated endocytosis of LDL particles. Okay. We've also discussed how synthesis of cholesterol can occur in these cells and how that is regulated as well. Okay, right. We then moved on to discuss what happens if these cells get an excess of cholesterol. So we discussed that the hepatocytes, which have an excess of cholesterol, are going to just make more bile acids, okay? They're going to convert the cholesterol into bile acids and then into bile salts. And we discussed that the peripheral cells, which have an excess of cholesterol, they can partake in this reverse cholesterol transport pathway in which they first give the cholesterol to HDL particles. The HDL particles turn this into cholesterol esters, which can then be put into LDL particles, and hence really the peripheral cells are putting cholesterol back into the LDL pool. Okay, what we now want to discuss is the final two pathways on this diagram, which are the exogenous pathway and the endogenous pathway. The exogenous pathway is the pathway by which cholesterol from the dietary cholesterol, can be delivered to the hepatocytes. Okay, now the exogenous pathway, really what we'll see when we study the exogenous pathway is that really the cholesterol delivery to the hepatocytes is not the main focus of the exogenous pathway. The main purpose of the exogenous pathway is actually to deliver triacylglycerols from the diet to the adipocytes and the muscle tissues. Okay, then the delivery of cholesterol to the hepatocytes is almost like an afterthought. Okay. It's similarly with the endogenous pathway, which is another pathway which will only actually be occurring in the fed state. What happens is it's the pathway by which the hepatocytes that are now overloaded with nutrients of all different kinds, not just cholesterol, okay? The hepatocytes which are overloaded with nutrients of many different kinds, so for instance glucose and amino acids, they can use this to synthesize triacylglycerols, and then the endogenous pathway is really about them delivering these triacylglycerols to the adipocytes and to muscle tissue. Okay, and then again, the delivery of cholesterol into the LDL pool is just almost an afterthought, basically. Right, so we're now going to discuss then these two exogenous and endogenous pathways, and I've warned you about how now the movement of cholesterol is almost like an extension on the side of the movement of triacylglycerols. Okay, right, so we're going to start off then by discussing the exogenous pathway, this pathway by which we can move cholesterol from the intestine, from enterocytes to the hepatocytes. But as I say, it is mainly a pathway about moving triacylglycerols from the enterocytes to the adipocytes and to muscle tissue. Okay, right, so the first thing that we need to discuss then is which lipoproteins are involved. We know that to move lipid molecules around the body, they need to be packaged as lipoproteins. Okay, we've seen LDL and HDL. The next one that we're going to talk about, because it's the one of the exogenous pathway, is chylomicrons. So the first thing I want to talk about, if we're going to talk about the exogenous pathway, is chylomicrons, and I want to do a more in-depth study of the chylomicrons. Okay, so when we went over lipoproteins in general, we said that chylomicrons were the giants of the lipoprotein world. Okay, so they are massive. They can have diameters from 70 nanometers to 1,200 nanometers, so they're big lipoproteins. Okay, the other fact that I gave at the time when we discussed lipoproteins in general were that the two important apolipoproteins in chylomicrons were apolipoprotein B48 and also apolipoprotein C2. Okay, and we will see that apolipoprotein B48 is again a giant apolipoprotein which has a function very similar to apolipoprotein B100 on the LDL particles. Okay, and we'll see also the function of apolipoprotein C2 later, which will have a new function that we haven't yet seen. Okay, right, so let's just draw a picture of our chylomicron here. Okay, so once again it's a great big sphere. Okay, and now it is massive, so this diameter could be up to 1,200 nanometers, so 75 to 1,200 nanometers. That means now that the phospholipid monolayer needs to be really thin, because we are seeing this picture hugely zoomed out, so the phospholipid monolayer should be really thin, like so. Okay, now apolipoprotein B48 is another giant apolipoprotein, like apolipoprotein B100. Okay, so it is a big apolipoprotein, but of course it doesn't look as big as apolipoprotein B100 did on the LDLs, because the LDLs were so much smaller than the chylomicrons. Okay, so this can represent our apolipoprotein B48 here in turquoise. And now let's put the other important apolipoprotein on here, which is apolipoprotein C2 over here, and I'll colour in apolipoprotein C2 in red. Now, as I said at the time when we were discussing lipoproteins in generality, the chylomicrons will have many other apolipoproteins beyond these two that I am telling you, but these are the two that I am actually going to tell you the function of, okay, and therefore these are the two essential ones that you need to know. Okay, if you want, you can go away and learn the full list of apolipoproteins that are present on chylomicrons, but these are the two that it's absolutely imperative to know. Okay, right. Now, the phospholipid monolayer is a normal phospholipid monolayer. It will mainly consist of lecithin molecules. There'll be a few cholesterol molecules dotted around. Okay, what's now important is what's in the lipid core. Okay, because most of the lipid core of chylomicrons is not cholesterol esters. The two lipoproteins that we've studied in detail so far, the HDL and the LDL particles, their core has been full of cholesterol esters. Okay, it's not the case for the chylomicrons. The main component of the lipid core of chylomicrons is not cholesterol esters, but instead is triacylglycerols. Okay, so there's the glycerol molecule in green, and here are the long chain carboxylic acids attached to that glycerol molecule there in orange. Okay, so I'll just label this up as a triacylglycerol tag molecule. Okay, so 90% of the massive great core of chylomicrons is full of triacylglycerols, basically. Okay, so they are the main thing which chylomicrons are delivering from the enterocyte to other tissues of the body. Okay, you will have some cholesterol esters in here, but they are rare compared to the triacylglycerols. They, as I said at the start of this video, are the afterthought, if you like, for chylomicrons. Okay, right. So chylomicrons are these massive, great lipoproteins which are full of triacylglycerols and which are going to be produced by the cells that line the intestine. Okay, so enterocytes. Now, I said when we were discussing HDL that I was going to tell you a little bit more about how cells actually produce these lipoproteins. Okay, so I'm going to tell you what is known. Okay, what is not known is how these things are assembled inside cells, but we do know the subcellular location where these things are assembled inside cells. We don't know how they're assembled in that location though. Okay, so let me just draw a little picture then of an enterocyte here. Okay, so here are the microvilli, at the apical surface of our enterocyte here, and this is its basolateral surface here. Okay, right, so let me just put the nucleus on here, and that's the inner nuclear membrane. Then we'll have the outer nuclear membrane, which will be continuous with the ER membrane, and I'm drawing the ER because that's the place where lipoproteins are believed to be produced. And in the enterocytes, and I'll just label this up as an enterocyte, we are going to be producing these massive, great Kylomicron lipoproteins. Okay, right, so I'll underline enterocyte in some nice colour. Okay, so here then is the endoplasmic reticulum, and in the endoplasmic reticulum of the enterocytes, you're going to be producing these chylomicrons, which I'm going to represent as a purple blob here. Okay, so the enterocytes, when you've just eaten a fatty meal, are going to be getting loads of triacylglycerols coming into their endoplasmic reticulum because they're going to be absorbing the triacylglycerols from the diet. Okay, now note, the triacylglycerols do not just get into the enterocyte intact. They firstly have to be broken down into glycerol and free fatty acids, but once they're in the enterocyte, they're reconstructed into triacylglycerols. Okay, so we've got lots of triacylglycerols within the enterocyte. We're also going to be getting a lot of cholesterol absorption into the enterocyte. Okay, so we've got a lot of cholesterol. We can build cholesterol esters from the cholesterol. Okay, we'll also be getting a lot of lecithin coming into the enterocyte because lecithin will be a large component of the diet. Okay, so we're getting all the components basically of the chylomicrons coming together. Okay, the cell will then obviously have to produce the apolipoproteins, and then somehow the whole thing assembles in the ER in ways that are not understood. Okay, then what's going to happen is this is just going to be secreted in the classical way that things are secreted. Okay, so if I add in the Golgi here, what's going to happen is you're going to pinch off a little vesicle from the ER here, okay, which will contain the chylomicron there in purple, and then this little vesicle is going to go to the Golgi. Okay, so the chylomicron will now end up inside the Golgi. It will then move to the later layers of the Golgi, the later cisternae, like so. And then from the Golgi you'll have another little secretory vesicle pinch off, which will contain the chylomicron there. So here's this other vesicle, and then that will fuse with the plasma membrane to release the chylomicron out onto the basolateral surface of the enterocyte. Now, the chylomicrons do not go into normal capillaries, okay, and this is important. If they went into normal capillaries, they would then go into normal veins, and they would end up having to go through the hepatic portal vein, and then they go to the liver first. Okay, they do not, however, follow that pathway. Instead, they are drained by lymphatic capillaries, special capillaries, and classically lymphatic capillaries are shown in turquoise, so I will stick to that. So this little line here, this can represent a lymphatic capillary. Okay, and intestinal cells, enterocytes, have a very good drainage by lymphatic capillaries. There are a lot of lymphatic capillaries basically supplying, or rather draining, enterocytes of the small intestine. Okay, right. So, where do all of these lymphatic capillaries drain into? Well, they drain into a structure that is called the Sternocylene. This is a structure that is so big that it is actually taught in anatomy lessons, okay? This isn't a histology lesson thing, this is an anatomy lesson, it's big. Okay, so I'll just remind you of a bit of anatomy then. Where is the Sternocylene? Okay, so the Sternocylene is this large sort of junction of loads of big lymphatic vessels all coming together, and it's in front of the first and second lumbar vertebrae. Okay, so this is what I'm drawing here. So this is the first lumbar vertebrae, and then this is the second lumbar vertebra. Okay, so L1 and L2, and then sitting in front of L1 and L2, you then have this bulge, and this is the Sternocylene, and it will have a vessel draining it that's going up, and we'll talk about what the name of that is in a moment. And it has 3 important vessels coming into it here. So here are 2, and then one's right on the front here. So let me now add a bit of colour onto this. So we'll start by talking about these 2 that are coming in from the side. So this one here and this one here. Okay, so these are what are known as the lumbar trunks, okay, and they have the lymphatic drainage that's coming from the two lower limbs. Okay, so you have a left lumbar trunk here and a right lumbar trunk here. Okay, then this one right down the middle here, which I'll colour in vivid purple, this is the one we're interested in. This is what's known as the intestinal trunk. Okay, and this is where all of these lymphatic capillaries that are draining the intestines, okay, and therefore which will be having the chylomicrons coming into them, they're all going to drain into this intestinal trunk. Whoops, and it should be intestinal trunk there. Okay, right, so I'll colour it in to hide my mistake. So here, intestinal trunk. Okay, so all of these lymphatic capillaries that drain enterocytes of the intestine are eventually going to be draining into this intestinal trunk, which will be draining into this massive great bulge that we've got here, sitting in front of L1 and L2, okay, which is easily visible, and this is called the Sternocylene, okay, like so. Then you'll have one big vessel draining the Sternocylene, okay, which I'll colour in red here, and this is known as the thoracic duct, okay, and this is the way that the chylomicrons, through draining in this lymphatic system, are going to avoid the liver, basically, okay, they're going to avoid that hepatic portal circulation, okay, because this thoracic duct is not going to go to the liver, basically, it's going to drain into the systemic venous circulation, okay, so this is the thoracic duct. Okay, right, so the thoracic duct is now going to go up the thoracic vertebrae, okay, and then it becomes asymmetric. At the moment everything's in the midline, but the thoracic duct is going to lean over to the left-hand side. So let me now show you where it is actually going to join the systemic venous circulation. Okay, so for this we need to draw the massive great veins that all join together to form the superior vena cava. Okay, so at the top of the thorax then, you have the superior vena cava, which will be going into the right atrium. Okay, so I will just label this up as the SVC here, which stands for, well, I should probably write it out in full. This is the superior, that's the S, and then the V is for vena, and then the C is for cava. Okay, and we'll colour in the superior vena cava in blue here. Now the superior vena cava is made from two vessels that are above it, okay? So I'll show you now these two vessels that are going to come together to make the superior vena cava. Okay, so a left vessel here and a right vessel here, and we're looking from the front, basically. Okay, so the heart will be here with the right atrium here. Okay, and this is what's above the heart, basically. Okay, right, so these two important vessels which come together to make the superior vena cava, these are known as the brachiocephalic veins. Okay, so you have a left brachiocephalic vein, which is the one I have pointed to here. Okay, and you also have a right brachiocephalic vein, which is this one over here. Okay, so let's just colour in the brachiocephalic veins in turquoise here. So this is the left one, and this is the right one. Okay, and of course we're talking about patient's left and patient's right. Okay, right, then the brachiocephalic veins, they are also made by the joining of two veins together. Okay, so let's now show this. So these are the last ones we're going to discuss. So here are the two that form the left brachiocephalic vein, and here are the two which form the right brachiocephalic vein. Okay, so what now are these called? So this one here, this is known as the internal jugular vein. Okay, and again you're going to have a left internal jugular vein and a right internal jugular vein here. So this is the internal jugular vein, and that's coming down from the head. Okay, so that is draining blood from the head and from the brain. Okay, so left internal jugular vein, or just the internal jugular vein. Alright, and then finally, this big vein here in orange, this is the subclavian vein, basically. So again, we'll label it up on the left-hand side here. So this is the left subclavian vein, and that's draining blood from the arm. Okay, right. So we are viewing this, I'll say it again, from the front. Okay, so this is the left side, this is the right side for the patient. Okay, and now I want to show you where the thoracic duct is actually going to drain into this system. So I'll show this in vivid purple. Okay, so I'm going to continue the thoracic duct on in vivid purple rather than red because I've used red for the internal jugular vein. Okay, so it continues up along the midline behind the superior vena cava, but then it starts to move over to the other side. I'm showing it in dashes because it's behind all of this. Okay, and then I can show it properly, okay, because it's not behind anything now, but then it goes behind that internal jugular vein, and then it will drain into the left subclavian vein here. So it's asymmetric, it doesn't run in the centre line. Okay, there is a sort of half-hearted attempt at something on the other side, okay, but that doesn't drain the intestine, so we won't mention it. Okay, so this is the thoracic duct then here. So it moves over onto the left-hand side and drains into the left subclavian vein there. Okay, so this means that these chylomicrons are going to come up in the thoracic duct, and they're then going to drain into the left subclavian vein, therefore into the superior vena cava, and then they'll go into the right atrium, into the right ventricle, they'll go around the pulmonary circulation, into the left atrium, into the left ventricle, and then they'll go around the systemic arterial circulation. Okay, and they will be delivered to peripheral tissue, so they have avoided the hepatic portal circulation is the key thing here. Okay. Right, so they do not go to the liver first. Instead, they go to skeletal muscle and cardiac muscle and also adipocytes. So remember, the chylomicrons, they're not really about cholesterol. They are about triacylglycerols. They have a huge number of triacylglycerols, and they need to find somewhere to deliver these triacylglycerols to. Now, you don't want to be delivering them to the liver. What's the liver going to do with them? Okay, send them on to something else. No, there's no point. We'll take them to the place where they actually are going to be used or stored. So that's what chylomicrons are now going to do. They're going to take them to skeletal and cardiac muscle tissue where they can actually be used for metabolism. So muscle tissue likes to use lipid molecules, fatty acids for metabolism rather than glucose. Okay, it prefers beta oxidation over glucose. Then you can also take them to adipocytes in adipose tissue where of course they can be stored for later use. Okay, right, so let's now talk about how we can then deliver these triacylglycerols to the skeletal and cardiac muscle cells and also to the adipocytes. Okay, right, so in the capillaries of these tissues, okay, I'll just write out these special tissues. So muscle tissue, both skeletal and cardiac muscle tissue, is going to be able to harvest the triacylglycerols from chylomicrons. Okay, and also adipose tissue is going to be able to harvest the triacylglycerols from chylomicrons. Okay, and in these two types of tissue here, the capillaries are going to have a special enzyme on their surface, which is how you're going to harvest the triacylglycerols from the chylomicrons. Okay, so let me show you an endothelial cell then. So here is the basement membrane of our capillary. Okay, and then we'll have an endothelial cell sitting on top of the basement membrane. Okay, so this is some endothelial cell within a capillary in one of these types of tissue here, so either in muscle tissue or adipose tissue. And basically in these special tissues where chylomicrons are actually going to be harvested, the endothelial cells of the capillaries are going to have a special enzyme on their surface. Okay, so let me put this enzyme here. Okay, so it's not an integral membrane protein, but it is on the surface of the cells. Okay, it's attached to other things that are on the surface of the cell, okay, which we won't go into, but the principle is what's important, which is that it's on the surface of the cell. Okay, facing into the lumen of the capillary, okay, where the chylomicrons are going to be. Okay, and this enzyme is really, really important. This is what's known as LPL. Okay, and this stands for lipoprotein, that's the LP. Okay, and then lipase. So lipoprotein lipase are a really important enzyme on the surface of these endothelial cells here. Okay, so I'll just underline it in some nice colour. Right, so what then is lipoprotein lipase going to do? Well, basically, when a chylomicron comes along, the lipoprotein lipase can stick itself into the chylomicron. Okay, it then gets activated by the apolipoprotein C2, and it can now break down triacylglycerols into glycerol and free fatty acids. Okay, so let me now show this. So I will draw my chylomicron here, okay, like so, and here is the phospholipid monolayer that makes up the outer aspect of the chylomicron. Here we'll have the apolipoprotein B48, and that's going to become important later on. Okay, so we'll see the purpose of this later. Okay, but now the star of the show is this apolipoprotein C2, okay, which binds and activates lipoprotein lipase here. Okay, so this is apolipoprotein C2 here in orange. Okay, so our chylomicron has just happened to come into a capillary in the skeletal or cardiac muscle tissues or the adipose tissues, and here on the surface of those endothelial cells of the capillaries you have this lipoprotein lipase enzyme which has stuck itself into the chylomicron and has then bound to the apolipoprotein C2, which now activates the lipoprotein lipase. Now what then does the lipoprotein lipase start to do? Well, it starts to work on the triacylglycerols that are in the core of our chylomicron here. So here is our triacylglycerol here. Okay, and what's going to happen is the lipoprotein lipase is going to break down those triacylglycerols into glycerol and then free fatty acids. Okay, so you're going to produce free fatty acids, okay, or just free fatty acids to be. Okay, so there is the glycerol molecule in green here. Okay, so that's glycerol, and then here are the free fatty acids, which are often abbreviated as FFAs. Okay, so this is for free, as in the on their own and not attached to something free, rather than as the number free. But of course, you do produce free fatty acids. Okay, right. So that's what lipoprotein lipase does. It breaks down triacylglycerols, which are the key cargo of these chylomicrons, into glycerol and free fatty acids. Okay, and these will then leave the chylomicron. They will be able to cross the endothelium and they'll go into the tissue. Okay, now, if they're going into skeletal muscle tissue, what's now going to happen is the skeletal or if they're going into skeletal or cardiac muscle tissue, then what's going to happen is the skeletal or cardiac muscle tissue will then just use the glycerol and the free fatty acids for metabolic purposes. So it will break them down and produce ATP from them, basically. Okay, so in the muscle tissue, what you'll use them for is metabolism. Okay, so principally beta oxidation is what we'll use those fatty acids for. Okay. In the adipose tissue, of course, something very different will happen. What you'll do is you'll reassemble the glycerol and the free fatty acids once they have arrived in the adipocyte. So I'll just draw this for the adipose cell. Okay, so here is our adipocyte here. Here's its nucleus. Okay, and then the adipocyte has a huge lipid droplet that dominates its cytoplasm. Okay, and basically once the glycerol and the free fatty acid molecules have come into the adipocyte, they will then be reassembled into triacylglycerols in the adipocyte, and they'll then be put into the lipid droplet of the adipocyte, okay, to be stored for later use, basically. So here is the glycerol molecule with the free long-chain carboxylic acids attached to it. Okay, right. So this is what chylomicrons do with their triacylglycerol cargo. They deliver it to tissues that can either use it, such as the skeletal and cardiac muscle tissues, or store it, such as the adipocytes. Okay, right. Now, what's then going to happen to the chylomicron is that it's going to gradually lose more and more of its triacylglycerols. Okay, so the core of the chylomicron here is going to get smaller and smaller and smaller as it loses more and more of its triacylglycerols, which it is giving away to these tissues here. Okay, now, as it gets smaller and smaller and smaller, then we need to lose some of this surface phospholipid monolayer, because otherwise what will end up happening is these chylomicrons will stop being spherical and they'll end up becoming more disc-like, like the HDL discs. Okay, if they want to remain spherical, they need to give away some of their surface phospholipid monolayer. Okay, and generally what they do is they give it away to HDL particles. Okay, so remember HDL particles, if I just bring this back up again, HDL particles, they really need phospholipids, okay, because they need the lecithin molecules to continue doing this reaction where they're adding acyl groups onto cholesterol molecules, okay, this reaction that's catalyzed by LCAT. So they're very quickly going to run out of lecithin molecules unless they're being given more, okay. So one of the places where they can get more is from these chylomicrons that are shrinking because the triacylglycerols are being taken out of them, okay. So HDLs can gain more phospholipids from the chylomicrons which are shrinking. Okay, they can also gain apolipoproteins from the chylomicrons that are shrinking. Okay, so these chylomicrons are also going to want to lose some of their apolipoproteins. Okay, and they can give them to HDL particles. Okay, then what will we end up with? We're going to end up with a much smaller chylomicron now, and I'll just draw this out here. Okay, so here now, this is our smaller chylomicron. Okay, this has lost a huge amount of the triacylglycerols from its core. Okay, and what now dominates the core, rather than it being the triacylglycerols, it's now the cholesterol esters, basically. Whoops. So here is the cholesterol molecule in red. Okay, and I'll just try and re-straighten up the camera. Okay, that's fine. And then we've got the acyl group attached to the cholesterol molecule here in orange. So these cholesterol esters are going to become a much bigger component of the lipid core of these chylomicrons as more and more triacylglycerols are removed. Okay, you've still got your massive great apolipoprotein B48 here, and we're soon going to see the function of that apolipoprotein B48. Okay. In addition, you've now got quite a bit of cholesterol in the membrane now, in the phospholipid monolayer. Okay, because you've given away lots of your phospholipids to the HDL particles, and therefore the cholesterol molecules have ended up becoming a lot more dense, basically, in that phospholipid monolayer. Okay, so this structure that we've now got left over here, this is what's known as a chylomicron remnant. Okay, it's what's left over of the chylomicron once it has performed its function of delivering the triacylglycerol from the intestine to the muscle tissue and the adipose tissue. Okay, now it contains a lot of cholesterol and cholesterol esters. Okay, and now it's going to carry out that function of delivering the cholesterol from the diet to the hepatocytes, basically. Okay, so the way that this occurs is through a similar mechanism to the way that the LDL molecules are absorbed into cells. Okay, so this is how apolipoprotein B48 here is going to be really important. Okay, because you remember that apolipoprotein B100 was really important for the endocytosis of LDL particles. So basically, chylomicron remnants can be endocytosed into hepatocytes in a very similar way to how we saw that LDL particles could be endocytosed into hepatocytes and peripheral tissues. Okay, and in fact, chylomicron remnants are able to bind to LDL receptors themselves. So apolipoprotein B48, like apolipoprotein B100, is capable of binding to the LDL receptors ligand binding domain. Okay, and therefore it can use the LDL receptor to be endocytosed. Okay, however, the main one that chylomicrons use is not the actual LDL receptor itself, but instead it is receptors very similar to the LDL receptor, which are a family known as the LRPs. Okay, so the LRPs are LDL receptor related proteins. Okay, so LDL, that's the L here. Okay, then the R is for receptor. Okay, then you have a dash related, so the related doesn't get a letter in the abbreviation, and then the P is for proteins. Okay, so LRPs are LDL receptor related proteins. Okay, so these are surface receptors that the liver cells will have on their surface, which are very similar to LDL receptors. Okay, they carry out very similar functions. Okay, and they can bind to apolipoprotein B48 better than the LDL receptor can bind to apolipoprotein B48. Okay, so remember the LDL receptor is principally good at binding to apolipoprotein B100 on the LDL particles. Okay, some of these LDL receptor related proteins will be better at binding to apolipoprotein B48 and taking up the chylomicrons from the blood through, again, a receptor-mediated endocytosis pathway. Okay, now, I should stress that there is not just one LRP receptor. There are loads of different LRP receptors, and many of them are capable of doing this function of endocytosing chylomicrons from the blood into hepatocytes. Okay, so overall what we have now seen then is that these chylomicrons are produced by enterocytes, which are absorbing nutrients from the lumen of the intestine. Okay, their first and principal job is delivering triacylglycerols to the adipose tissue and muscle tissue, so skeletal and cardiac muscle tissue. Then, once they've run out of triacylglycerols, they start to shrink. Okay, they lose phospholipids to HDL particles, they lose apolipoproteins to HDL particles, but they retain that apolipoprotein B48, and they become what's known as a chylomicron remnant, okay, which has a lot of cholesterol esters stored in that lipid core. It also has cholesterol in the phospholipid monolayer, and it now is going to deliver the dietary cholesterol to the hepatocytes, okay? And it is endocytosed into the hepatocytes in a mechanism similar to the LDL receptor endocytosing LDL, and indeed apolipoprotein B48 can bind to the LDL receptor on the surface of hepatocytes, but there are other receptors which can bind to chylomicron remnants, can bind to the apolipoprotein B48 in this family of LRPs.